In this module, we will talk about the pH control system while designing a fermenter. pH is very critical. All the biological systems require a specific pH. Minor change in a pH that controlled by the buffering capacity while designing the medium, because we add some buffer compounds which can control a minor change in a pH value. What is pH? We know that is the negative logarithm of hydrogen ion concentration, and that is some different ionic behavior of that. It's a chemical property of any system, of the medium. So if we set the pH at the start of the fermentation medium formation, suppose we set the medium at pH 7. So when we sterilize, sometimes at the result of the sterilization, there is a change in the pH. Sometimes when there is a growth, by the depletion of some specific ingredient, suppose as an example, when there is ammonium sulfate and the organisms utilize the sulfate ions, and then in the medium remain ammonia. So that's why that such factors has which can alter the pH. On the other hand, sometimes when the organism produce some product that have an acidic nature or they have alkaline nature, then they can tend to change the pH of the medium. Sometimes pH, we have to regulate the pH just for the sake of some desired product. In case of calcium gluconate, in case of sodium gluconate, we have to keep the pH at 7. So, because when there will be automatic run and there is no pH control, then there is a gradual change in the pH to the acidic side. So, the pH control system is very critical. Normally, when we want to control the pH, we have to use either the alkali agent or the acid agent. So, this alkali and then acid, then that's how much is the total system should be controlled. So, pH is very critical. So, as concerning what are the agent which can be utilized as a neutralizing agent. So, the neutralizing agent used to control the pH should be non-corrosive, because in case of the hydrochloric acid, if the body vessel is stainless steel, we cannot use any chlorinated compound, either those compounds which have INE in the end. So, we cannot use such compounds. So, they should be non-toxic to the cells when diluted in the medium. So, the potassium hydroxide is preferred in case of alkali, sodium hydroxide, as potassium ions tend to less toxic to the cells than sodium ions. So, KOH is more expensive than NOH. So, one is the type of the compound and other is the rate of that. So, we have to be very careful. So, here you can see that hydrochloric acid should never be used as it is corrosive even to the stainless steel. So, sulfuric acid in a concentrated should not be between 10% and 80%, as between this range of sulfuric acid is more corrosive. So, these are very strong compounds. We should be very careful while selecting our neutralizing agent. So, for laboratory fermenters, the peristaltic pump is used to add the pH adjusting agent, and then that is linked with the different kind of the tubing, and mostly the silicone tubing is used. So, the silicone tube will decay in the presence of high alkali concentration, and when we sterilize that material in a high temperature, then the silicone tubing have a restricted life. So, alternatively, we can have a choice of using the Tygon and neoprene tubing. So, the Tygon is not autoclaved but can be sterilized by passing the sodium hydroxide through the tubing for about 1 hour. And the neoprene is autoclavable but is not transparent. So, that's why the Tygone or the silicone tubing is mostly used for the addition of the neutralizing agent. So for fermentation that produce a large amount of acids, for example lactic acid, fermentation is using media, then there is a molarity, some specific molarity is needed. So alkali that have 4 molars and above is preferred. So this will prevent the dilution of the medium, because when we use the diluted neutralizing agent, that can raise the volume of the medium, that can affect the kinetic parameters. So we should use a specific strength and the concentration of acids or the alkali agents which we use as a neutralizing agent.